Every living organism is composed of chemicals which can be either elements or compounds. The elemental composition of living tissues can be determined by following a technique known as elemental analysis. Interestingly, we obtain similar elements while doing an elemental analysis of different living and non-living things. For example, we find that elements such as carbon, hydrogen, oxygen are common to the Earth's crust as well as living tissue. However, the composition of these elements is much higher in a living organism compared to the Earth's crust, as shown in this table. To ascertain the chemical composition of organic compounds in a living organism, we perform chemical analysis. For example, to find the chemical composition of a living tissue, such as a vegetable, we first grind it in trichloroacetic acid. As a result, we obtain a thick slurry. Now we strain this slurry through a cheesecloth to obtain two fractions. One fraction is the filtrate or the acid-soluble pool, and the other fraction is the retentate or acid-insoluble fraction. The acid-soluble pool contains organic compounds. Once a compound has been extracted, the next stage is to separate one compound from the others using separation techniques such as chromatography and centrifugation. Thereafter, various analytical techniques are used to identify the molecular formula and the structure of the compound. All the carbon compounds we derive from living tissues are called biomolecules. For example, carbohydrates, lipids, amino acids, water, and nucleic acid. Biomolecules such as amino acids and sugars are also known as metabolites because they are intermediates and products of metabolism. They are classified as primary and secondary metabolites. Compounds such as hydrogen, carbon, oxygen, nitrogen, sulfur, and sodium, which are involved in normal growth, development, and reproduction, are called primary metabolites. They are found in animal tissues. On the other hand, compounds such as alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, and spices are known as secondary metabolites. They are found in plant, fungal, and microbial cells and perform an important ecological function. Some of them play a helpful role as antibiotics and pigments. Living beings are also composed of inorganic compounds such as sodium chloride and calcium carbonate. To analyze the constituents of inorganic compounds, we carry out the following process. We take a small amount of living tissue, such as a leaf, and weigh it. This weight is known as wet weight. Then we dry this leaf to allow the water to evaporate to get its dry weight. Now, when we burn the leaf, the carbon compounds get oxidized and the leaf releases carbon dioxide and water vapor. The remaining ash contains inorganic elements such as calcium, magnesium, and potassium. Therefore, Analysis of compounds helps us identify organic and inorganic constituents in a living tissue. It also helps identify functional groups such as aldehydes, ketones, and aromatic compounds. In biology, however, functional groups are present as amino acids, fatty acids, and nucleotide bases, together known as biomolecules. Amino acids are the building blocks of all proteins. The amino group and carboxylic group are present on the same alpha carbon atom of the amino acid. Hence, they are also known as alpha amino acids. Amino acids are substituted methanes. There are 4 substituent groups occupying the 4 valency positions. These groups are hydrogen, the carboxyl group, amino group, and a variable group also known as the R group. Based on the nature of the R group, there are many amino acids. However, there are 20 standard amino acids in proteins. For example, glycine, in which the R group is hydrogen, alanine, where the R group is a methyl group, and serine, where the R group is a hydroxymethyl group. The chemical and physical properties of amino acids are essentially of the amino, carboxyl, and the R functional groups. As amino acids contain both the amino group and carboxyl group, they can be acidic, such as glutamic acid, basic, as in the case of lysine, and neutral, like valine. Similarly, there are aromatic amino acids such as tyrosine, phenylalanine, and tryptophan. A unique property of amino acids is the ionizable nature of the NH2 and COOH groups. Amino acids behave as both bases and acids. In solutions at a particular pH, ions with both positive and negative charges are equal. This form is known as the zwitterionic form. Another group of biomolecules is fatty acids. Long chains of fatty acids and their derivatives lead to the formation of lipids. Lipids include a broad range of molecules such as fats, waxes, fat-soluble vitamins, and glycerides. They are generally insoluble in water. Fatty acids that form lipids have a carboxyl group attached to an R group. The R group could be a methyl, ethyl, or a higher number of CH2 group with 1 carbon to 19 carbons. For example, palmitic acid has 16 carbons, including a carboxyl carbon. Fatty acids can be saturated or without a double bond. They can be unsaturated or with one or more C double bond C. Glycerol or trihydroxypropane is a simple lipid. When fatty acids combine with glycerol, they form esters, also known as glycerides. Glycerides can be monoglycerides, diglycerides, and triglycerides. Based on their melting points, glycerides may be either fats or oils. Oils have a lower melting point and hence remain as oil in winter, while fats freeze easily at low temperatures. Lipids, that contain phosphorus and a phosphorylated organic compound are known as phospholipids. They are found in the cell membrane. Living organisms also have many carbon compounds with heterocyclic rings, such as adenine, guanine, and thymine. These compounds are called nucleotide bases, and they are important biomolecules. When carbon compounds with heterocyclic rings are attached to a sugar, they are called nucleosides. For example, adenosine and thymidine. On the other hand, if a phosphate group is also found esterified to a sugar, they are called nucleotides such as adenylic acid and thymidyllic acid. Nucleic acids such as DNA and RNA consist of nucleotides. They are molecules involved in heredity. Thus, biomolecules act as building blocks of life and perform vital functions in living organisms. All living organisms produce organic molecules called biomolecules. Biomolecules can be broadly divided into two types: biomicromolecules and biomacromolecules. On performing a chemical analysis, we find that biomicromolecules are chemical compounds found in the acid-soluble pool with molecular weights ranging from 18 to 800 daltons. Some examples of biomicromolecules are water, minerals, amino acids, sugars, lipids, and nucleotides. On the other hand, biomacromolecules are compounds found in the acid-insoluble fraction. They have molecular weights in the range of 10,000 daltons and above. Biomacromolecules, or compounds which come from the insoluble fraction, are proteins, polysaccharides, and nucleic acids. Interestingly, lipids, which are biomicromolecules, are also found in this fraction. The acid-soluble fraction represents the cytoplasmic composition, while the acid-insoluble fraction consists of macromolecules derived from cytoplasm and organelles. Together, these two fractions represent the complete chemical composition of living organisms. Biomacromolecules have molecular weights in the range of 10,000 daltons and above. Let's understand why lipids, whose molecular weight does not exceed 800 daltons, are a part of the acid-insoluble fraction. Lipids have a small molecular weight, and they are arranged in the cell membrane and in the membranes of mitochondria, chloroplast, and Golgi apparatus. During a chemical analysis, when we grind a tissue, the cell structure ruptures and various membranes break into pieces and form vesicles. These vesicles are not water-soluble because lipids are hydrophobic. So, they get separated along with the acid-insoluble pool. We'll now learn about polysaccharides in the acid-insoluble fraction. Polysaccharides are long chains of sugars. They are thread-like structures consisting of different monosaccharides as building blocks. In a polysaccharide chain, for example in starch, the right end is known as the reducing end, while the left end is called the non-reducing end. Polysaccharides can be divided as homopolysaccharides or heteropolysaccharides. When all the monosaccharide units in a polysaccharide are of the same type, it is called a homopolysaccharide or simply homopolymer. Some examples are cellulose, starch, glycogen, and inulin. Cellulose is a homopolymer consisting of only glucose as a monosaccharide unit. Some examples of cellulose are a plant's cell wall, paper made from plant pulp, and cotton fibers. Starch, on the other hand, is a homopolymer of glucose which acts as the storehouse of energy in plant tissues. Starch forms helical secondary structures, and in that position it can hold iodine molecules. Conversely, cellulose cannot hold iodine molecules as it does not contain complex helices. In animals, glycogen acts as a homopolymer formed of glucose as a monosaccharide unit, a variant of starch. Inulin is a polymer of fructose. Polysaccharides, which are formed of more than one type of monosaccharide unit, are called heteropolysaccharides or simply heteropolymers. They are complex in nature, with amino sugars and chemically modified sugars as building blocks. Glucosamine and N-acetyl galactosamine are examples of heteropolymers. Another type of biomacromolecule in the acid-insoluble fraction of living tissues are nucleic acids. A nucleotide acts as a building block of nucleic acid and forms a polynucleotide chain. A nucleotide has three chemically distinct components, namely a heterocyclic compound, a monosaccharide, and phosphoric acid or phosphate. The skeletal heterocyclic ring compounds are actually nitrogenous bases such as adenine, guanine, uracil, cytosine, and thymine. There are two types of nitrogen-containing bases, namely purines and pyrimidines. In a purine, 6-member and 5-member nitrogen-containing rings are fused together. Pyrimidines, however, have only a 6-member nitrogen-containing ring. Therefore, adenine and guanine are substituted purines, while uracil, cytosine, and thymine are pyrimidines. The sugar found in nucleotides is either ribose or 2'-deoxyribose. Both these are monosaccharide pentose sugars. Therefore, nucleic acid containing deoxyribose is called deoxyribonucleic acid or DNA, while the one with ribose is called ribonucleic acid or RNA. Both DNA and RNA carry genetic information and are found in the cells of all living things. Together, polysaccharides, nucleic acids, and polypeptides or proteins form the macromolecular fraction of all living tissues. Proteins are important biomolecules needed by all living organisms. They are polymers of amino acids arranged in a linear chain and linked by peptide bonds. Therefore, they are also known as polypeptides. Amino acids that go into the making of proteins can be essential or non-essential. essential amino acids are found in our diet, while non-essential proteins are synthesized by our body. There are 20 types of amino acids that come together to form proteins, and so they are also known as heteropolymers. In a heteropolymer, there are different types of monomers repeating n number of times. Proteins form very interesting three-dimensional structures. The structure of molecules is depicted differently in different disciplines. In chemistry, structure means the molecular formula. For example, salt is also known as sodium chloride or NaCl, where NaCl is the molecular formula and not the structural formula. On the other hand, organic chemists present a two-dimensional view of molecular structure. For example, in benzene or naphthalene, this structure represents the structural formula, whereas the molecular formula is C6H6. Physics considers a three-dimensional view of molecular structure. However, biochemists have described the protein structure at 4 levels: primary, secondary, tertiary, and quaternary structure. Let's learn more about them. The primary structure is the simplest structure, which refers to the positional information of amino acids in a protein. Hypothetically, a protein is a thread formed by amino acids where the left end is represented by the first amino acid or the N-terminal amino acid, and the right end is represented by the last amino acid or the C-terminal amino acid. The protein thread is represented as a horizontal straight line so that the position of the amino acids can be determined. However, in nature, proteins are not completely straight but are folded in the form of a helix, somewhat like a revolving staircase. In the secondary structure, amino acid links get longer, and due to the heaviness of the molecule, it tends to bend. This preliminary bending is considered to be the secondary structure. It is either an alpha helix or beta sheets. The third level of protein structure is the tertiary structure. It refers to the three-dimensional structure of the entire polypeptide chain. The function of a protein, except as food, depends on its tertiary structure. If this is disrupted, the protein is said to be denatured and it loses its activity. The fourth level of protein structure is the quaternary structure. This structure is formed by several protein molecules or polypeptides, also known as protein subunits. These subunits are arranged as a linear string of spheres arranged one upon each other in the form of a cube or plate. For example, adult hemoglobin consists of 4 subunits, 2 of which are identical to each other. Therefore, 2 alpha-type subunits and 2 beta-type subunits collectively form human hemoglobin. Some of the most common proteins we come across are collagen, rubisco, trypsin, insulin, antibodies, receptors, and GLUT4. Let's learn more about each of these proteins. Collagen is the most commonly occurring protein in the animal world. Collagen fiber is generally found in connective tissues and acts as an intercellular ground substance in the space between the cells. On the other hand, ribulose bisphosphate carboxylase oxygenase, or Rubisco, is the most abundant protein in the whole of the biosphere. It acts as a catalyst during the process of carbon fixation. Trypsin acts as an enzyme, and insulin acts as a hormone. Another protein called antibody helps fight infections. Receptor, another protein, helps in sensory reception, while glucose transporter 4 or GLUT4 helps in transporting glucose to the cells. In this manner, various proteins play a vital role for every living organism. The structure of these proteins is crucial in determining their functions. Bonding plays a crucial role in the making of a polymer. A polymer is formed when many monomers come together and bond in long chains. For example, polyethylene is made up of repetitive units of ethylene monomer. Let's look at the bonding in biomolecules such as proteins, polysaccharides, and nucleic acids. Proteins are amino acids linked by peptide bonds. This chemical bond is formed when the carboxyl group of an amino acid reacts with the amino group of the next amino acid, losing a water molecule. This process is also known as dehydration. Polysaccharides are formed when several monosaccharides link together by a glycosidic bond. This bond is also formed due to dehydration. The glycosidic bond is a functional group that can exist between two carbohydrate molecules or between a carbohydrate and a non-carbohydrate molecule, as between glucose and serine. The bonding in nucleic acids is different. Here, the 3' carbon atoms of one sugar of a nucleotide are linked to the 5' carbon atoms of the sugar of the succeeding nucleotide by a phosphate moiety. The phosphate and hydroxyl groups of sugar join to form an ester bond. The ester bonds formed by the phosphate group between the 3rd and 5th carbon atoms of adjacent pentose sugar rings are called phosphodiester bonds. This bond links 3' carbon atoms of one sugar molecule with the 5' carbon atoms of an adjacent sugar molecule. The phosphodiester bond is very vital to all forms of life, as it forms the backbone of deoxyribonucleic acid or DNA strands. According to the famous Watson-Crick model, the B form of DNA is the native conformation of DNA in solution. The B form of DNA is a metabolically stable configuration. Some of the features of bDNA are that it exists as a double helix. Two strands of polynucleotides run in opposite directions. In other words, they are antiparallel. That is, one strand runs in 3' to 5' and the other strand runs in 5' to 3' directions. The backbone of DNA is formed by a sugar-phosphate sugar chain. The nitrogen bases face inside and are almost perpendicular to the backbone. The 4 bases found in DNA are adenine, cytosine, guanine, and thymine, which are abbreviated as A, C, G, and T respectively. Each type of base on one strand forms a bond with just one type of base on the other strand. For example, adenine on one strand base pairs with thymine on the other strand by a double bond. Similarly, cytosine on one strand base pairs with guanine on the other strand by a triple bond. Each strand appears like a helical staircase, and every step of ascent is represented by a pair of bases. One full 360-degree turn of the helical strand would involve 10 steps or 10 base pairs. In other words, there are 10 phosphate sugar groups in every full revolution of the DNA strand. The distance along the helix axis corresponding to one turn is known as pitch. In a B DNA, the pitch is 34 angstroms. The rise per base pair, or the distance between two base pairs, is 3.4 angstroms. The different types of bonds we have studied are crucial as they provide structure and stability to biomolecules and make them functional. All living organisms, whether plants, animals, or microorganisms, contain innumerable organic compounds or biomolecules in a certain concentration. This concentration is expressed as moles per cell or moles per liter. Interestingly, biomolecules constantly break up and are remade, or in other words, they have a turnover. They change into other biomolecules or are made from other biomolecules due to constant chemical reactions in living beings. The entire set of biochemical reactions that takes place in living organisms and helps them maintain life is known as metabolism. Metabolism is important to living beings to maintain their structures, growth and reproduction, and helps them respond to their environments. However, metabolic reactions lead to the transformation of biomolecules. For example, the decarboxylation of histidine gives histamine. Similarly, the hydrolysis of a glycosidic bond in sucrose yields glucose and fructose. Most of these reactions do not occur in isolation and are usually linked to other reactions. In fact, the series of linked chemical reactions that transforms biomolecules is called metabolic pathways. These pathways take a specific path and include specific biomolecules. They can be either linear, as in the process of glycolysis, or circular, as in the Krebs cycle. Metabolic pathways can be divided into two categories: anabolic pathways and catabolic pathways. Anabolic or biosynthetic pathways lead to more complex structures from a simple one. For example, the process of glycogenesis, which involves the biosynthesis of glycogen from glucose. In other words, glucose molecules are added to chains of glycogen for storage. On the other hand, catabolic pathways lead to a simple structure from a complex one. They involve degradation and also lead to a release of energy. For example, the process of glycogenolysis, which leads to the breakdown of glycogen to glucose. The energy released during degradation is trapped by living organisms and stored as chemical bonds. Later, this bond energy is utilized for biosynthetic osmotic, and mechanical work whenever the need arises. The most important biomolecule that contains energy in its chemical bonds is adenosine triphosphate, or ATP. When bonds of ATP break, a lot of energy is released. ATP also transports chemical energy within cells for metabolism. All the chemical reactions associated with metabolic pathways are catalyzed reactions. Metabolic pathways can be compared to automobile traffic, where they have traffic junctions when they meet and cross each other. Just like automobile traffic, the flow of metabolites through these pathways is definite and takes place in a specific direction. This flow of metabolites is also called the dynamic state of body constituents. As you have studied, thousands of metabolites or biomolecules are present in living organisms. Each of these biomolecules has a definite concentration. For example, the blood concentration of glucose in a normal healthy individual is 4.5 to 5.0 millimolars, while that of hormones is measured in nanograms and milliliters. Moreover, these biomolecules are constantly a part of the metabolic process. Any chemical or physical process moves naturally to equilibrium. However, systems at equilibrium cannot perform work. Therefore, the living state is a steady state or a non-equilibrium state, which is essential for living organisms to be able to work constantly. The living state or non-equilibrium state is attained by energy input through metabolism. Hence, metabolism and the living state are synonymous, and one cannot exist in the absence of the other. Did you know that cheese is produced with the help of rennet, which is a natural complex of many enzymes? Enzymes act as biological catalysts in a living system and change the rate of reactions without themselves getting altered. While most enzymes are proteins, some nucleic acids also behave like enzymes. These nucleic acids are called ribozymes. Just as with proteins, enzymes have a primary, secondary, and tertiary structure. While the primary structure is an amino acid sequence, the tertiary structure folds and crisscrosses on itself, creating many crevices or pockets. One of these pockets is called the active site. A substrate fits into this active site of the enzyme and enables it to catalyze reactions at a high rate. While enzymes catalyze chemical reactions, they are very different from inorganic catalysts. Inorganic catalysts such as platinum and nickel work effectively at high temperatures and under high pressure, while enzymes are damaged at temperatures above 40 degrees centigrade. However, some enzymes, such as thermolysin, can be isolated from thermophilic organisms that live in high-temperature environments like sulfur springs. These enzymes retain their catalytic power even at temperatures as high as 80 to 90 degrees centigrade. To understand the working of enzymes, we'll begin by learning more about chemical reactions. Chemical compounds undergo physical and chemical changes. When a compound changes shape without breaking its existing bonds, it is said to be a physical change. For example, ice melting to form water. On the other hand, when the bonds in a compound are broken and new bonds are formed during transformation, it is called a chemical reaction. The rate of a reaction is defined as the amount of a product formed per unit time. It can be expressed as delta P upon delta T. Various factors such as temperature and pressure influence the rate of reaction. This rate generally doubles or decreases by half for every 10-degree change in temperature in either direction. For example, consider that a particular reaction is taking place at 5 degrees with the rate of reaction as 1 unit. When the temperature is increased by 10 degrees, the reaction rate will double to 2 units. Similarly, if the temperature is decreased by 10 degrees, the reaction rate will become half. A catalyst greatly influences the rate of a reaction. Hence, the rate of enzyme-catalyzed reactions is significantly higher than uncatalyzed reactions. Consider the example of a reaction where carbon dioxide and water react to form carbonic acid. In the absence of any enzyme, 200 molecules of carbonic acid are formed in an hour. However, when the enzyme carbonic anhydrase is used, 600,000 molecules of carbonic acid are formed every second. Hence, the carbonic anhydrase enzyme accelerates the reaction 10 million times. There are numerous types of enzymes, and each of them catalyzes a unique chemical and metabolic reaction. There are also multi-step reactions, where each step is catalyzed by a single or multiple enzymes. For example, glucose is converted into pyruvic acid through 10 different enzyme-catalyzed metabolic reactions. Pyruvic acid yields different types of end products during various pathways. During aerobic respiration, it forms acetyl-CoA and carbon dioxide in the presence of the pyruvate dehydrogenase enzyme. While during anaerobic respiration in the muscles, in the presence of the enzyme lactate dehydrogenase, pyruvic acid leads to the formation of lactic acid. On anaerobic respiration in yeast, the same pyruvic acid leads to alcoholic fermentation in the presence of the enzyme pyruvic decarboxylase. Therefore, metabolic pathways catalyzed by enzymes yield different types of metabolic end products under different conditions. Enzymes are three-dimensional protein structures with an active site. They convert a chemical or a substrate denoted as S into a product or P. One or more products can be formed from a single or many substrates. Enzymes help in catalyzing the reactions, but they do not participate in the reaction themselves. In other words, at the end of any reaction, the enzyme remains unaltered and is ready to be used by another molecule. The multi-step reaction mechanism involving enzymes as catalysts is called the catalytic cycle. We shall now study the steps in this cycle. First, the substrate S binds to the active site of the enzyme and fits itself inside a given cleft or pocket. In the second step, the binding of the substrate forces the enzyme to change its shape and fit more tightly around the substrate. This leads to the formation of a highly reactive enzyme-substrate complex or ES complex. This complex is short-lived due to its instability, but it is extremely important for catalysis. These temporarily formed complexes are also known as transition state structures. In the third step, the active site of the enzyme breaks or forms chemical bonds, and a new enzyme product complex or EP complex is formed. This EP complex is also unstable. Finally, in the fourth step, the structure of the substrate is transformed into the structure of the products. During the enzyme action, there can be many temporary, unstable structural states between the stable substrate and the product. For example, ES and EP complexes. These states are also known as altered structural states. Stability refers to the energy status of the molecule or the structure. In other words, stable structures can exist in nature without any uptake or release of energy. For example, S and P have a low energy status and are stable, whereas ES and EP complexes with a high energy status are unstable. This is a graph which shows how enzymes lower the activation energy during reactions, thereby increasing the rate of reaction. Activation energy is the minimum amount of energy required for the chemical reaction to take place. In other words, it is the minimum amount of energy required by the substrates to form products. The y-axis represents the potential energy content, while the x-axis represents the formation of products via complex structures. Notice that there is an energy level difference between S and P. If P is at a lower level than S, it implies an exothermic reaction releasing heat energy. In other words, an external supply of heat energy is not required during this reaction to form a product. However, whether an exothermic or an endothermic reaction, the S has to go through a much higher energy state or transition state. In the graph, the red curve indicates that in the absence of an enzyme, the activation energy for a reaction is high. On the other hand, the pink curve shows that in the presence of an enzyme, the activation energy gets lowered, due to which the substrate transforms into a product quicker. Therefore, enzymes reduce the activation energy barrier by making the transition from S to P easier. In this way, enzymes bring about chemical and metabolic conversions at a high rate. Enzymes are mostly proteins, and they increase the rate of chemical reactions. Just like proteins, enzymes also have a primary, secondary, and tertiary structure. A change in a few factors can affect the activity of enzymes and can alter the tertiary structure of the protein. These factors include temperature, pH, change in substrate concentration, or the binding of specific chemicals that regulate the activity of enzymes. Let's take a look at each of these factors. Temperature and the pH factor affect the enzyme activity immensely. Typically, enzymes function in a narrow range of temperature and pH. While low temperature preserves the enzyme when it is in a temporarily inactive state, high temperature destroys the activity of enzymes as the proteins get denatured by heat. In other words, the tertiary structure of proteins is destroyed and they become functionally inactive. Every enzyme demonstrates its highest activity at a particular pH level and temperature, which are called optimum pH and optimum temperature respectively. These following graphs depict the impact of change in pH and temperature on enzyme activity. As shown by the graphs, enzyme activity increases steadily with an increase in pH and temperature up to the optimum pH and optimum temperature. Enzyme activity is the highest at this point. Thereafter, with an increase in pH and temperature, enzyme activity decreases as they get denatured and are not available for catalyzing reactions. Notice that enzyme activity is also low below the optimum value. Change in substrate concentration is the second factor impacting enzymatic activity. Here's a graph which shows enzyme velocity at many different concentrations of substrate. If the substrate concentration increases, initially, the velocity of enzymatic reaction rises. In the final stage, the reaction reaches a maximum velocity or Vmax. This velocity does not rise any further, even if there is an increase in the concentration of the substrate. The reason is that at Vmax, enzyme molecules are less than substrate molecules. After the saturation of these molecules, there are no free enzyme molecules that can bind with the extra substrate molecules. The third factor that shuts off enzyme activity is the binding of a certain chemical. This process is called inhibition, and the chemical that induces it is called an inhibitor. For example, malonate is a competitive inhibitor of succinate, which gets converted to fumarate in the presence of succinic dehydrogenase. Sometimes the molecular structure of the inhibitor closely resembles that of a substrate. Such an inhibitor is known as a competitive inhibitor, and it impacts the activity of the enzyme. Since this inhibitor is structurally similar to the substrate, it competes with the substrate for the substrate binding site of the enzyme. As a result, the substrate cannot bind and enzyme action is reduced. For example, penicillin binds at the active site and inhibits the transpeptidase enzyme that cross-links the peptidoglycan strand to form the bacterial cell wall. This competitive inhibition is used to control bacterial pathogens. Thus, enzyme activity depends on physical factors such as temperature and pH, and the chemical surroundings such as substrate concentration and the binding of specific chemicals. Enzymes play a significant role by helping the various processes in a biological cell to take place at a significant rate. Scientists have discovered and studied numerous enzymes. Depending on the types of reactions they catalyze, enzymes have been divided into 6 classes, namely oxidoreductases or dehydrogenases, transferases, hydrolases, lyases, isomerases, and ligases. Each of these classes is further divided into 4 to 13 subclasses, named by a 4-digit number or EC number. In an EC number, for example, A, B, C, D, A will denote the class, B the subclass, C the sub-subclass, while D will denote the sub-sub-subclass. Oxidoreductases or dehydrogenases are enzymes that catalyze the transfer of electrons and hydrogen ions from one molecule, called the reductant, to another molecule or the oxidant. Here, the reductant is the electron donor, while the oxidant is the electron acceptor. An example of an oxidoreductase enzyme is lactic dehydrogenase, which converts pyruvic acid into lactic acid and vice versa. The second class of enzymes is transferases. These enzymes catalyze the transfer of a functional group G, other than hydrogen, between the two substrates S and S'. For example, the enzyme transmenase transfers the amino group from alanine to oxaloacetic acid, forming pyruvic acid and aspartic acid. The third class of enzymes, or hydrolases, catalyzes the hydrolysis of bonds such as ester, ether, peptide, glycoside, carbon-carbon bonds, halide bonds, or phosphorus and nitrogen bonds. For example, the hydrolysis of glycosidic bonds in sucrose by sucrase leads to the formation of glucose and fructose. Lyases comprise the fourth class of enzymes that catalyze the removal of bonds by mechanisms other than hydrolysis and oxidation. Lyases use mechanisms such as forming double bonds or a new ring structure for catalysis. An example is aldolases, which cleaves the aldol groups in fructose 1,6-biphosphate and forms dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The fifth class of enzymes, called isomerases, catalyzes the interconversion of optical, positional, and geometric isomers. For example, during glycolysis, phosphohexose isomerase catalyzes the conversion of glucose 6-phosphate to fructose 6-phosphate. Ligases form the sixth class of enzymes. They catalyze the linking together of two compounds. For example, the joining of carbon with oxygen, carbon with sulfur, carbon with nitrogen, and phosphate with oxygen bonds is catalyzed by ligases. As you know, enzymes are three-dimensional protein structures joined by polypeptide chains. However, sometimes non-protein chemical compounds also bind to the enzyme and make it catalytically active. These chemical compounds are called cofactors, while the protein part of such enzymes is known as apoenzyme. There are three kinds of cofactors: prosthetic groups, coenzymes, and metal ions. Prosthetic groups are organic compounds that are tightly bound to the apoenzyme. For example, in the enzyme complex succinate dehydrogenase, FAD of flavin-adenine is a prosthetic group that oxidizes succinate to fumarate in the 8th step of the citric acid cycle. coenzymes are also organic compounds which get bound to apoenzymes for a brief period during catalysis. In fact, coenzymes act as cofactors in a number of different enzymatic reactions. Common examples of coenzymes are NAD or nicotinamide, adenine, dinucleotide, and coenzyme A. The third type of cofactors is metal ions. They are required by enzymes to form coordination bonds with side chains at the active site and one or more coordination bond with the substrate. For example, magnesium is a cofactor for glucose 6-phosphatase and manganese is a cofactor for arginase. Some commonly occurring metal ions are cobalt, manganese, zinc, and copper. A cofactor is very crucial for the catalytic activity of the enzyme. If the cofactor is removed, the enzyme loses its ability to perform catalysis. Therefore, the classification of enzymes provides information about reactions carried out by different enzymes.